Okay, good morning, everyone. So in case some of you are struggling in this course or with the immunological principles that are presented, there is tutoring through the academic success programs, and there are tutors for immunology available. So please check with them if you think that you need some help. So where we left off last time were the initial steps in T cell activation. And I've sort of broken it down into sort of proximal events, right, which is the initial recruitment of LCK and FYN, the kinases that phosphorylate the CD3 zeta chains of the T cell receptor. And that recruits ZAP70, which phosphorylates the linker of activation in T cells. That recruits other kinases. And then you sort of get this cascade, okay? And so I don't think that you need to know all of the steps, okay? What I do think you need to know are the things that I've highlighted on the right, okay? And so, and even this is a very simplified version of the intracellular signaling pathways. But the main steps are sort of, how do you get the activation of the different transcription factors? And so the first one is the phospholipase C gamma is going to cleave phosphoinositol diphosphate to give you IP3 and diacylglycerol. And these are both second messengers. So IP3 is going to open up the calcium stores. Calcium gives you NFAT-C activation. That's a transcription factor. Diacylglycerol will activate protein kinase C theta that will phosphorylate the inhibitor of NF-kappa B to generate active NF-kappa B. So those are two transcription factors. And then the MAP kinase program will lead to phosphorylation of phos and Jun, which together form the AP1 transcription factor. And what you might not have noticed when I put this up last time is, what is it turning on? And the thing that's getting transcribed here, among other genes, is a cytokine called interleukin-2, okay? This is a key T cell growth factor. If you remember, Kendall Smith originally cloned this gene, and it was initially called T cell growth factor. We now call it interleukin-2. And the reason that this is important is because interleukin-2 will cause the T cells to start proliferating. And so what happens is that naive T cells express only the gamma and beta subunits of the IL-2 receptor. And so that's a low to moderate affinity receptor for interleukin-2. But once these T cells are activated, they're going to make IL-2 and the alpha chain of the IL-2 receptor. Okay, this causes the IL-2 receptor to become higher affinity for IL-2. It's about 100-fold higher affinity. This allows the T cells to respond to the cytokine and start proliferating. Okay, so the first step after T cells get activated, they start to proliferate. And when they're doing that, they're going to differentiate into effector T cells. Now, the dogma is that IL-2 is coming from CD4 cells. But in reality, CD8 T cells will make some IL-2. But without CD4 help, that's not enough IL-2 to sustain them, and they'll eventually die off. So activated CD4 T cells make much more IL-2. And that's what's really causing, if we ever talk about CD4 help for CD8 cells, it's really just IL-2 production. And so what's happening here is a resting T cell expresses only the moderate affinity IL-2 receptor, or the low affinity version, okay? And then as it's activated, those signaling cascades are happening, you get activation of NFAT, NF-kappa B, and AP1. You get transcription of IL-2 and the IL-2 receptor alpha chain. So they're making both the cytokine and the high affinity version of the cytokine receptor. And so what happens is that, at least initially, the T cells are making cytokines that they use themselves. And this is called autocrine IL-2, as opposed to paracrine, which is one cell making it for the cell next to it. This, at least for CD8 T cells, is sufficient to give them this initial burst of proliferation. Okay, so when you're thinking about initial steps in T cell activation, you have CD4 and CD8 T cells, and they are recognizing different MHC and different peptides. And so they may recognize the antigen on the same APC, or it may be different APC, okay? And this activation occurs independently, but the main help for CD4 T cells to CD8 T cells is production of IL-2. Now there are other signals like CD40 ligand is again used to activate T cells, but it's mainly the production of IL-2, okay? That allows the CD8 T cells to keep going. Now for both CD4 and CD8, the main result of activation is proliferation. Whenever these cells proliferate, they're differentiating into effector T cells. Now, if you've ever heard CD8, CD4 inversion as an indicator of an immune response, what that says is that CD8 T cells are proliferating much more than CD4 T cells. And so you get more CD8 cells than CD4s, and that's suggestive of that there is a response going on. So from last time, the questions about T cell immunity, or how does a T cell get activated, really boil down to how does the antigen get to the lymph nodes? And so you have migratory dendritic cells that will pick up antigen in the periphery, move to the lymph node, and then present this to T cells. Or if it's a small antigen, it's going to come in through the lymphatics, through the follicular reticular network, which are the leaky pipes, and lymph node resident dendritic cells will pick that up and present it to T cells. And then I think you should understand how do T cells enter lymph nodes so that they can encounter those antigen presenting cells. And so you should know the role of the high endothelial venules. What does CD62L or L-selectin do? And what is the role of CCR7 in activating LFA1 for those cells to get into the lymph node by the high endothelial venules? And then finally, what are the signals that an APC, an antigen presenting cell, is going to provide to T cells for activation? So you should know what is the immunologic synapse, right? This is where you push out those large proteins to the periphery that have the phosphatases attached to their cytoplasm, or their cytoplasmic tails, leaving the TCR signaling proteins on the center with the kinases. And so the phosphatases are pushed out, they can't inhibit the kinases, and that's how you get the signal. You should know what the three signals for the T cell are: the antigen signal, the co-stimulatory signal, CD28, and the cytokine signal, which is, in this case, IL-2. And then I am going to ask you questions about what is the signaling pathway in T cells, and really it boils down to that color-coded TCR signaling pathway. So are there any questions on that before we move on to CD8 T cell responses? So there's a question about phospholipase C-gamma. There are other phospholipases that are active in T cells, but C-gamma is the main one. And I think it gets a little bit superfluous to sort of cover all of the different signaling components. I think we just know the main pathways for this class, and then we'll go over some of the other ones in the advanced immunology course, but that's the main one. Okay. So let me get out of this. Okay. Sorry, going as fast as I can here. Okay, so now we're going to focus first on the CD8 T cell response because it's much simpler, right? The CD8 T cells have one job: get activated, leave the lymph node, and go and kill infected cells. They don't provide help for B cells. They're just the killers. So they're very simplistic. And so when we talk about an antigen a T-dependent B cell response, we have this idea, right, that the antigen is going to come in through the afferent lymphatics, either on macrophages or through the lymphatics if it's small, get transferred to follicular dendritic cells. The antigen-specific B cell will then recognize both the antigen and IC3B by complement receptor 2. And then it's going to go towards the T cell zone, okay? And if there's if there's no CD4 help early during response, or if this is not a protein antigen, then it's just going to keep going and it's going to form the primary foci, which are the short-lived plasmablasts. Mostly they're going to make low affinity pentameric IgM that aids in further complement activation, sort of reinforcing this antigen IC3B so you get more B cell activation. However, if there is CD4 help in the form of CD40 ligand signaling to CD40 on the B cells, instead of forming the primary foci, it forms the germinal center. This is where you have somatic hypermutation, affinity maturation, and isotype switching, and eventually generation of long-lived memory cells and plasma cells that make antibody. But when we're talking about T cells, you have sort of these two different ways, right? You have peripheral dendritic cells that are picking up antigen, and they're activated by PAMP receptors, and that tells them to go to a lymph node. They'll actually enter the lymph node and go all the way to the paracortex. Remember, they're following, excuse me, they're following the CCR7 signals, just like the T cells. That's going to do, so they're following CCL21 that's binding to CCR7. So they're moving towards the paracortex where the T cells are coming in. And the T cells are being activated either on the peripheral dendritic cells or on lymph node resident dendritic cells that have picked up antigen and are now presenting it to T cells. And so what happens is you get proliferation and differentiation to become effector cells, right? This is the whole idea. Those cells, those T cells get activated. They start making IL-2, and that allows them to proliferate and survive. And then they have to go do their job. And if we're talking about CD8 T cells, it's just exit to the blood, okay? So CD8 T cells don't provide help to B cells. They're just going to leave. But if it's CD4 T cells, right, they're going to provide help for B cells and also do other jobs. And we'll cover that in the next lecture, which is more about how do CD4 cells differentiate. But for this one, for this lecture, we're just going to talk about CD8 T cells and how they get activated and then what they do. Okay, so remember, we have this 3-signal paradigm of T cell activation. There's an antigen-specific signal, which is the TCR, and that is just recognizing, is this a foreign antigen? So that involves the T cell receptor and the co-receptor, CD4 or CD8. There's a co-stimulatory signal. This is the check to make sure that this antigen presenting cell is actually activated. So the T cell is going to express CD28. If the antigen presenting cell has gotten a PAMP receptor signal, it's going to express B7, which is the ligand for CD28. And so that's the co-stimulatory signal. That's, yes, this is something you should make a response to. Now the final signal is a cytokine, and this can be many different cytokine signals, but in reality, you need something to tell those cells that, yes, this T cell should proliferate. And the main cytokine that we've talked about is IL-2. I know it's not on this diagram, it's a little bit older, but IL-2 is the main cytokine that's telling those T cells proliferate and differentiate. Other cytokines, including IL-12 and TGF-beta, modulate that T cell, in particular for CD4 cells, to tell them what kind of CD4 cell to be. Okay, so there's a proliferation and differentiation signal, but there's also a modulatory signal that tells them, okay, do you want to make a IgE response or an IgG response? Now, in vitro, you can force this by providing very strong signal 1 and 2, okay? That is something that happens in a tissue culture, but doesn't really happen in vivo, except during some bacterial infections where you have superantigen. The exception to this, right, is that if there are strong TLR signals in the lymph node, the immune system sort of says, okay, the infection's gotten here, throw everything at it, everybody's gets the signal to respond. And that's just because if there's an infection in your lymph node, you've got to make any kind of response that you can. Okay, so here's our signaling cascade. I won't go over this again, but remember, I just want you to focus on the things on the right side of this to sort of get an overview of TCR signaling. But let's focus on what a T cell does, okay? And this is particularly important for CD8 T cells because a lot of people get confused. The initial signals for a T cell is antigen recognition and co-stimulation, and then cytokine production, okay? Now that, in particular for CD8 T cells, that's enough. That the T cell will proliferate and differentiate into what we call a cytotoxic T lymphocyte, or CTL. You may have heard that term. Many people confuse that by saying CTL and CD8 interchangeably. They're not. A CTL is a particular type of T cell that's been activated and is ready to go kill. So that initial activation doesn't cause the CD8 T cell to kill the dendritic cell. That would be counterproductive. What you want it to do is get activated and then proliferate on its own. And Sukhak and others have shown once the T cell gets activated, it just divides, undergoes program division, 7 or 8 divisions to give you a lot of T cells, but not killing the dendritic cell. Now that's different than once it's differentiated to become an effector T cell, or a CTL, then it's going to go out and start killing other cells. And here it's looking at, this is a B cell, and it's sort of saying, okay, well, what is the B cells presenting here? You can think of this as now a target cell. Once that cell has been activated, any cell it recognizes, it's going to kill. Okay, so that's the difference between activation and effector recognition. Now, effector T cells have these various steps, right? We've kind of covered these already. Here, a T cell is recognizing antigen presenting cell. So we have TCR recognition, there's CD8, which is this dimer, and it's also getting co-stimulation. So the CD28 and B7 recognition. That then leads to these transcription factors getting activated, and the T cell making the high affinity form of the IL-2 receptor and making IL-2. Or if there are CD4 cells nearby, those cells will make IL-2 that then can act on that T cell. And it's going to proliferate and differentiate, but then it leaves the lymph node. And once it goes out from the lymph node, then it's going to start this program of looking for infected cells. So it's not recirculating, it's actually going through and looking for signals to exit the blood and enter into tissues to start killing infected cells. Okay, now at this point, it doesn't require co-stimulatory signals. Okay, it's already programmed, you're a killer. So you don't need all of these accessory signals to say, yes, this cell in fact is infected. Because if that was the case, then a virus would just stop, would block expression of co-stimulatory proteins, and T cells wouldn't be able to kill it. Okay, so this kill signal doesn't require additional co-stimulatory signals. Now the dendritic cell is going to activate both CD4 T cells and CD8 T cells, but this sort of goes both ways, right? And the key step here is that CD4 cells are going to get activated. So they're going to have antigen recognition. They're going to get co-stimulation. They'll get cytokines, but they're providing a signal to the antigen presenting cell as well. Okay, so it's reciprocal. CD4 cells are getting activated, but they're also providing signals to the antigen presenting cell. What happens is that antigen presenting cell will now upregulate costimulatory proteins. And so this is called licensing, where CD4 T cells license antigen presenting cells to go ahead, activate as many CD8 T cells as possible. The reciprocal signal to the CD4 cell is it starts making IL-2 that is going to help activate CD8 cells, but then it's also going to start expressing CD40 ligand. Okay, and this is that key signal for CD4 cells to go help B cells, but it's also a key signal for CD4 cells interacting with CD8 cells in addition to making the IL-2. Okay, now there are a lot of other co-stimulatory proteins that we do not cover in this class. They mainly help shape the response in terms of what kind of helper cells do you want, how does that influence the CD8 cell, but we don't really cover those. And so here's sort of a general picture of a number of the other co-stimulatory molecules. There are ones that are inhibitory. The most well known is probably CTLA4. This is going to block CD28 signals. But once that T cell becomes activated, the CD28 signal binds to lots of B7, or what's also known as CD80 and CD86 on the antigen presenting cell. So if there's enough of a signal, it overcomes that blockade. The other ones are things like CD40 ligand on the T cell. This is going to then provide other signals to the antigen presenting cell. It's going to help for B cells and CD8 T cells. But that sort of CD28 leads to CD40 ligand. The other ones that are going to get expressed really are modulatory ones. OX40, 41BB, CD30 and CD153, and ICOS. I don't expect you to know all of these other ones, but things like OX40 is important for the type of T cells. So if OX40s get signaled by the antigen presenting cell, you have mostly a Th2 response. If you get 41BB, this is more for modulating B cell responses. Some of the other ones like ICOS and CD30 are going to modulate the type of T cells. So really the ones that I want you to focus on are CD28, right? That's the initial co-stimulatory signal, which is opposed by CTLA4. And so again, you have to overcome the negative signals to get the positive signal. You have to exclude the phosphatases, and that's how CD28 gets activated. If that gets activated, then the T cell will upregulate CD40 ligand, okay? Those are the three main ones. Okay, so now let's just focus on effector CD8 T cells and what they do once they get activated, okay? And they have, whenever you think about CD8 T cells, they're pretty monolithic. They don't have a lot of accessory jobs. They're simpler than CD4 cells. They just have one job, let's go out and kill infected cells or tumors or whatever the problem is. And once they get activated, they proliferate and differentiate into effector T cells, or sometimes called cytotoxic T lymphocytes. So the naive T cells, a naive CD8 T cell is going to enter the lymph nodes by the high endothelial venule. This is just a review. So they're going to express L-selectin, right, which is this low affinity lectin that binds to sialyl X carbohydrates in the HEV. And then they're going to express CCR7, which binds to CCL21, the chemokine, and that's going to activate LFA1, the integrin that allows these cells to do tight binding and get into the lymph node. You have the migratory dendritic cells. They're going to bring antigen in through the lymphatics, or the resident dendritic cells that are sitting there that will pick up antigen from the follicular reticular network and sit in the T cell zones and present it to T cells. And so this initial step of T cell activation for CD8 T cells occurs when the dendritic cell with the correct antigen MHC are recognized by T cells with the correct T cell receptor. Okay, so they're coming in different ways, but they're meeting in the paracortical region, and that's where activation occurs. Now for CD8 T cells, that's the whole story. They get activated there and then they leave. So if they come in, here are the green ones, they try to recognize antigen, but they don't see anything, they're going to downregulate CCR7, which is the chemokine receptor that says stay in the lymph node, and upregulate the S1P1 receptor, which says exit the lymph node. And so they go out through this actually should be medullary sinus, and eventually they go through lymphatics to get to the thoracic duct and re-enter the bloodstream. But the blue ones that come in, here are antigen specific, are going to recognize antigen on the dendritic cell, whether it was migratory or lymph node resident, and they become activated and start proliferating. And so they're making autocrine IL-2, they're getting some IL-2 from CD4 cells, but they're going to exit now. Okay, so the naive T cells come in and they exit if they don't recognize antigen. But if antigen recognition does occur, you don't want those cells to leave. So they don't upregulate S1P1 receptor. They tightly adhere to the dendritic cell, and they're going to start proliferating. Okay, so that requires these T cells to have tight binding to APC so you can get all of those recognition events happening. Now what happens then is that you want these cells to exit immediately into the periphery. If they were to go through the medullary sinus, as this would indicate, that takes too long. And so instead of going here, they're actually going out through the high endothelial venules. Okay, that's the direct path to the blood so they can get out and do their job the fastest. So this T cells, or these T cells that have been activated, they've got all the co-stimulatory signals and the cytokines that allow them to proliferate in the lymph nodes. During that proliferation, they differentiate into effector cells, and then they exit the lymph node to the blood, and their job is to go and fight the infection. And that means recognizing infected cells and killing them. Okay, now the process is the problem is now they're in the blood. How do they know where to go? They've gotten to the blood, but let's say the infection is in your big toe. How do they know to only go out of the blood into your tissues in the big toe? And the answer is the same process of following chemokines. Okay, so in this case, it's showing LTB4, which is not necessarily a chemokine, but it will bind to a receptor on the T cells. And it's the same process where the cells are sort of rolling through the blood vessel. They get a chemokine receptor, which causes activation of an integrin, and that then allows them to enter tissues. So in this case, here's our bloodstream. Okay, the cells floating around, and they're going through the blood. Here's our endothelial wall of the blood vessel, and here's our infected tissues. Okay, so it's sort of upside down from most of the time we think about this. The rolling and sticking is again mediated by selectins. If you remember, what is the selectin that allows the T cells to enter the high endothelial venule? Anybody chime in? See, we're all sleepy this morning. We covered the entry to the high endothelial venules. The rolling and sticking part is L-selectin. The L stands for lymph node. In the periphery, when you're rolling and sticking along blood vessels, you actually have the P and E selectins. This is platelet selectin or endothelial selectin. They have different jobs, and we'll cover that in a minute. But that's just slowing it down. The first step is that then you have activation. Okay, so that is a chemokine binding to the chemokine receptor, which causes the integrin to go from a low affinity form to a high affinity form. The integrin here, what they call it the activated integrin, now is a high affinity version and can bind tightly to ligands like ICAMs, intracellular adhesion molecules. that allows this to have firm adhesion and exit from the blood into the inflamed tissues. So it's a lot like how the cells get into the high endothelial venules, but it's a different selectin, it's different chemokines, then it actually uses some different integrins as well. Okay, so I should point out that there are some changes in the T cells as they become activated in the lymph node. They start changing, first of all, their selectins. Okay, to get into a lymph node, a naive cell wants L-selectin, but in general, once the T cells are activated and they leave the lymph node, you don't want them going back to the lymph node, you want them to enter inflamed tissues. So instead of expressing L-selectin, it expresses the ligands for other carbohydrates, and these are the P and E selectins. It will also upregulate the integrins. Okay, integrins are these dimeric proteins that go from a low affinity to a high affinity form. And why does it do that? Well, because other blood vessels are not like a high endothelial venule. They're not always low pressure. They don't have high surface area. So you have to upregulate the adhesion molecules so it's easier for cells to get out of the blood and into inflamed tissues. Now, those are the main ones. They change their selectins and they change, the main ones I want you to know are the changes in selectins and in the integrin, in particular LFA1. There are some other changes, in particular for human cells, they change the form of the adhesion molecule CD45RA to a different one. This allows more efficient activation. And they also change a marker, particularly in mouse cells, called CD44. This is just an adhesion molecule that allows cells to bind to other cells more efficiently. So the main ones are the selectins, the integrins, and then some of the other adhesion molecules, particularly 44 and 45. Okay? Now this is oversimplified. There are lots of other changes, but these are the main surface adhesion molecules that you really should know about, or that allow T cells to really stick to other cells so they can kill them. So there's a couple of issues here. One is you want to make this process more efficient. You want the cells to sort of know where to go. And so this is actually controlled by where the dendritic cells came from. If the dendritic cells are coming from the skin, you want them to inform the T cell that you should go back to the skin, okay? If they're coming from a mucosal surface like the lung, then you want them to inform the T cells to go back to the mucosal surface, in this case, the lung. Here, this is the gut epithelia, and then you're informing the dendritic cells, informing the T cell, yeah, you're gonna go back into the gut. And what that does is it actually changes some of the chemokine receptors that they're expressing so that they know which one to look for, and makes it more selective. And the reason for this is, if you think about it, just activate T cells and you don't tell them what they should be looking for, you'd have to express all of the chemokine receptors, and it's just not a very efficient process. So by doing this, the dendritic cells are saying, I got activated and I came from the skin, so you're gonna be looking for a skin homing signal. Or if it came from the gut, from a mucosal surface, it's going to say, I came from the mucosal surface, so you should be going back to a mucosal surface. Okay, so usually what happens is the T cells activated by a dendritic cell from a particular type of tissue will narrow what the T cell should be looking for. And it's basically saying, go back to where I came from, that's where the infection probably is. So gut dendritic cells will induce expression of alpha-4 beta-7 integrins. That's a different set of integrins that tells the cells you should be looking to go to the gut. They express a different chemokine receptor. And this allows them to know this is something that is typically expressed in gut inflamed tissues, sort of narrows down what they should be looking for. Okay, so this just makes the whole process more efficient. Do you have to know all of the different integrins? No, okay? but I'm just giving a few examples. Now, how this is controlled, it's mainly your immune system is going to treat your epithelium at the skin differently than the epithelium at mucosal surfaces. And this is controlled primarily by two vitamins. There's vitamin A, which is taken up as retinol, or retinoic acid. It's dehydrated from retinol to retinoic acid. Or it's taken up as the beta carotenes. Okay, if you've ever wondered what the heck beta carotenes are doing, their main job is actually influencing immune cells to go to mucosal surfaces. And so the dendritic cells that are from the mucosal surfaces are taking up retinol and converting it to retinoic acid that they then give to the T cells. And this is, retinoic acid will bind to the retinoic acid receptor, which is a transcription factor, and tells these cells you should go back to a mucosal surface. And so it'll express alpha-4 beta-7 integrin, CCR9, as a program to say, yes, I should be looking for an infection in a mucosal surface. The other vitamin is a closely related vitamin called vitamin D, right? And if you've ever, if you've lived in a very cloudy place like Seattle or London, people often take vitamin D supplements because there's not a lot of sunlight. Vitamin D is converted by skin cells to the active form, the 126 hydroxyl form. And so a dendritic cell in the skin will take up vitamin D and convert it to the active form and give it to the T cell. This will then bind to the vitamin D receptor, which is a transcription factor, and tells those T cells you should be looking for an infection in the skin, not in the mucosa. You should be looking for a skin infection, okay? And so it doesn't express alpha-4 beta-7, it expresses other integrins, and it expresses different chemokine receptors. It just narrows down where the T cell should be looking to go. Excuse me. Okay, so when we look at these different signals, it's essentially the same pattern, okay? And I'm just gonna highlight some differences. Here we have a naive T cell, and its job is to go from blood to lymph nodes. And so in the high endothelial venules, it's got L-selectin, which is going to bind to sialyl Lewis X. That slows it down to the rolling and sticking. It's got CCR7, which is going to recognize CCL21 on the high endothelial venules, and that's going to activate LFA1 to bind to ICAM1. So that's, you've got a rolling and sticking, you've got a chemokine receptor activation, and then tight adhesion. And it's just going to follow that chemokine gradient into the lymph node. Now, if it's gotten activated by a gut dendritic cell, or mucosal dendritic cell, then it's going to activate different things. So it's going to have P-selectin here. This is a different name for, or it's going to activate actually the ligand for P-selectin, which is expressed on the endothelial cells of the blood vessel. And so it's looking, this is the rolling and sticking step. Okay, so it looks a little reversed. There's a reason for that. It's easier to upregulate the selectins on the endothelial cells rather than on the T cells. But it's upregulating a different chemokine receptor, okay? And there's a number of chemokines that can activate it, but then you have a different integrin, okay? So there is a rolling and sticking by P-selectin. There's an activation step through CCR9. That results in tight binding by the alpha-4 beta-7 integrin. Okay, so it's the same steps, just different signals or different proteins doing this step. Now, if it came from the skin, you have, again, the main one is E-selectin, but P-selectin is also there. P stands for platelet. This is how platelets actually stick to endothelial cells. The main one is endothelium. And so it's got E-selectin, that is the rolling and sticking step. That's going to give you different chemokine receptors, okay? Main ones being CCR4. And that then results in activation of integrins. In this case, the alpha 4 beta 1 or LFA1. Okay, so the steps are the same. There's just different proteins involved at each step. And this is the initial activation tells that cell which ones it should be looking for, and then it goes out and responds to them. Okay, so the homing receptors are not enough. Once it's in the peripheral tissues, it's going to be sticking to target cells. And if there's no T cell receptor signals, then the cell will sort of move on to the next. And there are a lot of complicated steps in that, and I'm not going to require you to know all of those. But it's essentially the same migration process. The cells are going to keep looking in that inflamed tissue until they find an infected cell. So essentially what's happening, if there's no TCR signal, then the LFA1 goes to the low affinity form and it releases from that cell. Okay, so it stops the type binding. But instead, what happens if there is a TCR signal, so here's a TCR, it's recognizing the infected cell, that then is going to cause the LFA1 to stay activated, or if it's in a mucosal surface, the other integrins, stay activated and bind tightly to that cell and then kill it, okay? So this is continued activation of integrins. And then we'll talk about how it actually kills it in just a second. Now this organizes the T cell. And so it reorganizes as a microtubule organizing center so that it's basically shooting the cytotoxic gun at the target cell and not killing the other cells around it. So recognition and tight binding directs everything to come to that one infected cell. Once it kills that cell, then there's nothing for the LFA1 to bind to, right? It's a killed cell, so it's going to stop expressing the ligands, and that releases the T cell, so it can leave and then go and kill another cell. So you can sort of think of T cells as serial killers. We used to think that T cells would kill the target cell, but they'd also kill themselves or sort of have this fratricide problem. But that's not really how it works. It's really only killing the recognized cell. So a T cell can come into an inflamed tissue, can kill this cell. As that cell dies, it loses the adhesion molecules. The T cell will move to the next cell, recognizes that cell, kills it. As that cell dies, it loses the adhesion molecules, and the T cell will go to the next cell. Okay, and it can keep killing. Now this killing is a directed process, as I said. So the initial recognition, here's our CTL, our effector CD8 T cell, recognizes the target cell, and if there's TCR recognition, it reorganizes the microtubule organizing center, right, which tells the cell polarity, like how things should exit and enter. And that redistributes preformed cytotoxic granules, okay? These are the ones these are exocytic pathway. They are preformed killer granules. And this organizes everything so that it's only going to go out towards the target cell. Once you release those granules, they're going to kill the infected cell. Okay, that's the main mechanism of CD8 killing. So here's our cell. It's going to recognize the infected cell, and the red here are the cytotoxic granules. One of the components is a protein called granzyme B. And it's going to redistribute those to just that point of contact between the target cell and the CD8 T cell. The T here is target. Okay, and when it releases those are then going to have effects on the target cell to kill it. Okay, so this is really pointing the gun at the target cell. It's not just sort of exploding like a neutrophil. Okay, so how does it kill a target? How does a T cell kill a target cell? Well, the main thing that it's going to release are proteins called the perforin and granzymes. Okay, perforin is a granzyme transporter. Granzyme is the protein that actually kills the target. These are held in the T cell by a scaffold protein called cerglycan. Cerglycan sort of protects the T cell from killing itself, right? So if you're thinking of this in terms of gun, this is the bullet, but you don't want the bullet to fire backwards. So the T cell keeps the perforin and granzyme inactivated in the cytotoxic granules in the T cell by having them bound to cerglycan. Okay, now once it's released, those proteins are released from cerglycan and they go and do their job. Okay, so cerglycan is the scaffold that is which holds these proteins so they don't kill the T cell. Perforin forms a transporter. It was initially thought to be a pore forming subunit, like for example C9 in the complement pathway, but it doesn't. It forms a transporter. And then granzyme B, which initiates apoptosis in the target cell. Okay, so those are the three main proteins. So here's cerglycan, right? This is the scaffold protein, and it basically has these highly negative charged glycans stretching out from it, which will bind to granzyme B and to perforin, sort of hold them and protect them from forming pores in the exocytic vesicles. So this is the way it protects the T cell from killing itself. So if you just hold them, they can't form the transporter in the membrane of the cytotoxic granule inside the T cell. Can only do that when it's released. Now this is a lectin, so it binds to glycosylation sites on the other proteins. And it has lots of other roles as a scaffold protein and sort of holding things, preformed things that need to be distributed to other cells. And so it's active in neurons and other cells that have second messengers. But in T cells, its main job is to protect the T cell from killing itself. It holds these killer proteins in an inactive form, and so you don't get suicide, T cell suicide. Now, how it releases these is not exactly known. Once it sees the target cell and releases the granzyme B and perforin, they get released from cerglycan, and it's not clear if that's a pH system or if there's some other way that the proteins are released from cerglycan. But in any case, they are. So the first step is perforin is released from cerglycan, okay? And perforin will insert into the target membrane and to form a transporter unit. It's a pore-like structure where this is where it gets its name, is that they initially thought it was perforating the target cell, but it's a pore-like structure, but it doesn't cause membrane potential loss. It forms a transporter, and specifically it's a granzyme B transporter. And that's its only job, is to insert into the membrane and transport granzyme B, which is also released from cerglycan, into the target cell. Now, granzyme B is the main effector molecule in killing a target cell. Okay, so the CTL recognizes the target cell, it reorganizes all of its secretion apparatus, right? This is the Golgi microtubule organizing center, points the gun at the target cell, and it releases granzyme B and perforin bound to cerglycan. Those then, cerglycan releases granzyme B and perforin. Perforin inserts into the membrane, right here, and forms this perforin forms the granzyme B transporter, and that gets inside the cytoplasm. Now, granzyme B is going to do a number of things. Okay, the first one is going to activate caspase 3. Caspases are cysteine aspartate proteases. That's where they get their name. Caspases are made as a pro-enzyme, right? They're often called procaspase because it's inactive. Granzyme B will come in and cleave procaspase 3 into the active caspase 3 form. It will also come in and cleave BID. Okay, this is another protein that regulates mitochondrial electron transport. We'll go into those in just a second. But it's really just activating the cell's own death activation machinery, or apoptosis pathway. Okay, so the two things that are happening, right, is that you get activation of caspase 3. That leads to caspase 3 going on to cleave other proteins. And the main thing that's doing is it's cutting the inhibitor of caspase activated DNase, or CAD, which is, so ICAD is the inhibitory part of CAD, and releases that, and now that can go to the nucleus and starts chewing up DNA. It's basically cutting in between histones. So that's a bad thing for that cell, right? It's DNA fragmentation, so now you can't get, if you have that, you can't get transcription, and so it's shutting the cell down. The other thing that's happening is granzyme B is activating BID, and that's going to disrupt the mitochondria. In particular, it's going to cause release of cytochrome C from the electron transport chain. If you remember from biochemistry, if you don't have cytochrome C, you don't get any ATP, and that cell is shutting down. Okay, so caspase 3 is going to degrade the inhibitory subunit of CAD, caspase activated DNase. BID causes a release of cytochrome C, so now you've shut the cell down at the transcription level and at the energetics level. Okay, so those are the two main things that are happening, is just telling the cell, nope, you don't get to make any more proteins and you don't have any ATP, so you should just shut down. Now there are some variations on this. So if you just relied on this, a virus could potentially inhibit this in many different steps. So you'd have different ways of activating this. And the way that I think you should think about it is sort of, it's a sort of the backup plan. Granzyme B, right, this thing that comes from the T cell and enters the cytoplasm of the target cell, can directly activate caspase 3 to the active form. Okay, that's the main pathway. And then, as I just said, it can also directly degrade the inhibitory subunit of CAD to give you the active form of the DNase. Okay, so those are the pathways we just described. But there's some backup plans for this, right? There is, if you take BID and it cleaves it, that then will go to the mitochondria and you get release of cytochrome C. This, when it's released, will activate a second pathway, which is you'll get activation of caspase 9. Essentially what that does, it reinforces the caspase 3 pathway. Okay, so you can get caspase 3 from this way, or from BID activation and release, you get caspase 9, which then will cleave caspase 3. That's the backup plan. But now if, let's say you're a virus, well, why not just inhibit this? And then what's the backup plan? Well, granzyme B can also activate caspase 8, okay? And that can activate caspase 3. So there's 3 different ways. Granzyme B can activate caspase 3 directly, or through BID, give you cytochrome C, which activates caspase 9, or it can activate caspase 8, that then activates caspase 3. So it's direct through caspase 9 or caspase 8. Okay, so those are sort of the two backup plans, the main plan and then the 2 backup plans. The other thing that can happen, right, is that you have loss of cytochrome C, but that can then also release other enzymes that can cause DNA damage. Right? The mitochondria is a pretty toxic environment, and release of a lot of those components lead to cell death independently of this pathway. Now, as you might guess, there are a whole lot of caspases, right? I've only talked about 3, caspase 3, 8, and 9, but there are other ones. And there are lots of other ways, lots of other backup plans, but I don't think that going into all those details is really necessary. These are the main ones. Okay, a second way that cells kill. So this is activating apoptosis. There are other death pathways that we don't cover in this class called, this is apoptosis. This is sort of the safe way to kill a cell. There are unsafe ways. So if you don't have any of the activation of the caspases, you can get other things like necrotosis or pyrotosis, which are sort of immunologically dangerous, right? Because they actually allow you to make other responses that are potentially self-reactive. And we don't really cover those, but there are other pathways. The necrotosis pathway in particular is very important in some viral infections, but we're just going to focus on apoptosis. So how do we know this is important? Well, first of all, if you don't have this granzyme B pathway, then the host is susceptible to virus infection. And this was first shown with influenza infection. This is the mouse-adapted purate strain of influenza. And if you infect wild-type mice, they can control the infection and they survive, okay? But if you take mice that have been intentionally disrupted for the granzyme B gene and you infect them with influenza, they can't control it and they'll die, okay? And so that tells you, yes, it's important, okay? So it's essential for the control of many viral infections. But what people also noticed is that granzyme B deficient mice can still kill tumors and some viral infections. And so they're not susceptible to other things. And that conclusion from that was there must be alternative pathways for T cells to kill target cells. It can't just be perforin granzyme. We now know that there are other granzymes. There are granzyme A and others that can partially compensate for this, but there are also surface receptors that can kill target cells. And so what we now know is that there are proteins like CD95L or FAS ligand, or the TNF receptors that can bind to surface receptors on target cells and cause this apoptosis pathway. So here we have a T cell, right? T cell, or this also is true for NK cells. And they're going to have recognition and release of perforin and granzyme. The perforin transports the granzyme into the target cell. You get activation of caspase 3, activation of BID, and that leads to all of those events that we just talked about. Okay, this is sort of a simplified diagram where it's really focusing on, you get activation of BID, release of cytochrome C, that causes activation of caspase 9, and then downstream, you get caspase 3 and ICAD cleavage and DNA fragmentation. But these death receptors, the two main ones are FAS, or the FAS pathway, and the TNF receptor pathway. I don't care that you know all of the different TNF receptors. All I want you to really remember is there's death receptors, FAS ligand and TNF. Those are going to bind to these receptors on the target cell and initiate the same kinds of pathways. And so here, you can see the death receptors have procaspase 8 associated with them. And so once that binds, if you have T cells making the death signals, binding to the death receptors on the target cell, those then will result in procaspase 8 being released and activated to caspase 8. Remember, it's the same thing. Caspase 8 activates procaspase 3 to the active form. That then will cleave ICAD and you get DNA fragmentation, and that is killing the target cell. So the death receptors can still initiate the target pathway. And so if we go back here, really, if you have this pathway, this really comes from the death receptors. And so it's a backup plan. If you don't have granzyme B, well, you can't start this, right? But you can start this. So that's sort of why we include them in the same diagram, but they're really coming from two different receptor pathways.
Dr. Blattman?
Yes.
Do both the CD95 ligand and the trail ligand both need to bind at the same time in order to induce this pathway?
No, either one of them can do it separately. And so they actually have, for the FAS protein, this is FAS ligand binding to FAS. It uses an adapter called fast associated death domain protein, or FAD. For the TNF receptor, it actually has a sort of intermediate called TNF receptor associated death domain that will hold procaspase 8. So they use slightly different adapter proteins. They don't have to both bind. They can both independently cause cell death. We'll go over that in just a second. Now, so why does it use, how does it know to use these different pathways? Well, the way to think about this is if it's a viral infection or something that has lots of antigen, then it really does the perforin granzyme pathway. And that's just, it's much more efficient. So it's sort of like the T cell saying there's lots of antigen there. I can just use my best bullets to kill the target cell. But if there's low antigen, which occurs in some tumors or some viral infections, you know, for example, herpes viruses downregulate everything when they go to this latency phase. There's very little antigen, so the T cell is going to say, well, I'm going to use the death receptor pathway. Okay, so this sort of is a low antigen stimulus. You do get tumor cell death, but it's basically reserving its best bullets for something that's really immunogenic. But essentially the same thing is happening. So here's FAS ligand on the T cell binding to FAS or CD95 on the target cell. And then in the inside of that, it's already got, this diagram is a little incorrect. It's already got the FAS-associated death domain proteins holding procaspase 8. That gets released and cleaves procaspase 3 to the active form. Caspase 3 cleaves ICAD, the inhibitor of the caspase-activated DNase. That goes to the nucleus and starts chewing up the DNA. Okay, so it's the same steps, just a different way of starting it. So why do we know this is important? Well, you can do the same kinds of experiments, but instead of influenza, you infect mice with something that causes a lot less antigen. And as I said, herpes viruses will typically become latent, and so it's hard to detect them. There's very little antigen. And so if you take mice that are deficient in FAS, they cannot detect a low level of antigen. Wild type mice, the T cells will still be able to use this death receptor domain to recognize the low antigen level during herpes virus infection. So they control the infection, they survive. You use a FAS deficient mouse, so now the T cells can't see, and there's very little antigen, so the T cells can't use that alternative killing pathway. You don't control the herpes virus infection, and those mice will die. Now, which one's more important? Well, for normal, most of the time, the perforin granzyme pathway is usually the dominant pathway. That's the most important for most viral infections and tumors. The backup plan is sort of when special exceptions, when there's very little antigen there. Okay, so the death receptor pathway is more of a backup plan compared to the perforin granzyme pathway, which is very potent and important for killing cells. So how long does this take? Well, it turns out that cytolysis is pretty fast. So here we have an initial recognition of a T cell here. Here's our CD8 T cell and a infected cell, which I'll call I. Okay. In fact, there's actually two different T cells here recognizing it. But here you see that there are preformed granules, but they're pointed in the wrong direction. And so that T cell is going to bind to the target cell. It's going to have TCR MHC interactions, which are going to say, yes, this is your infected cell. And you can say very quickly, within a minute or within minutes, these preformed granules are getting pointed at the infected cell. Okay, then here they're in red. And within a few more minutes, within certainly an hour, but usually we think of this, it takes about a half an hour to kill a target cell. And you can see that here's our T cell, it's degranulated, releasing perforin and granzyme that are bound to seroglycin. And you can see this cell already, compare this cell to this cell, this is what looks like an apoptotic cell. It's starting to bleb its membrane, it's fragmenting its nucleus, that cell is done, okay? And so the T cell is going to release that cell and go to another cell. So within minutes, you reorganize the Golgi microtubule organizing center to point the bullet at the target cell. And within 5 minutes, you've degranulated and started this apoptosis pathway in the target cell. Apoptosis pathway takes 30 minutes or so for you to cleave up the DNA, release cytochrome C from the mitochondria and shut down that target cell. And now that T cell is ready to go on and kill another cell. What we do know that cytolysis is specific, that it's not just exploding. Remember, we go back to neutrophils, they recognize foreign things and they just sort of explode, releasing DNA nets. T cells don't do that. They're going to kill specifically infected cells and then move on. And if it's not infected, you don't want to kill it. Why? Because if you just start killing cells willy-nilly, there's too much collateral damage and that would lead to really immune pathology. Okay, so whether it's by the perforin granzyme pathway or by the Fas ligand, Fas pathways, it's really specific recognition of cells that need to be killed. You do this, you get CAD activation and DNA fragmentation, and you get release of cytochrome C, so you shut down the energetics. But it's not killing the neighbor cells. Okay, so unless they're infected, there's no reason to kill them off, so you preserve them and move on to something else. Now, something that we should know is that, or the way that we know this is something called an in vivo CTL killing assay. And this was developed by Dan Barber in Rafi Ahmed's lab, where you're injecting target cells. And so these are either target cells that have the peptide on them that T cells can recognize, or that don't have a peptide that T cells recognize. You inject those both into wild type mice, and then you look for killing of just the ones that have the peptide on them. So you can see that this peak gets killed and it goes away. But this one that doesn't have the antigen doesn't get killed, right? It's essentially preserved. And so from this, we know that T cells are only recognizing infected cells, or in this case, peptide pulse target cells, but not unpulsed cells. So it's not just exploding like a neutrophil. The other way that we know this is if you have influenza, or if you infect mice with influenza, you isolate those T cells, you can develop viruses that have lost that one epitope that is recognized by the T cells, okay? So you infect target cells, one that has the antigen, and target cells that are still infected, but the virus no longer has that epitope. And the T cells will only kill the infected cells that have the antigen. Even if they're infected, but they don't have the epitope to present on MHC, then they're not going to present anything to T cells and they won't be killed. Okay, so it's really the TCR recognition of antigen that controls whether or not that cell sticks to the target cell, polarizes itself, and releases the killing machinery. Okay, now just to mention this other pathway, right? We sort of focused on Fas ligand, But the TNF pathways are a little more complicated, and you don't have to know all the components here. But what I would like you to notice is that one, there's multiple different TNF family members. Okay, so cytotoxic T lymphocytes will make two main cytokines. They make tumor necrosis factor, and this is a whole family of proteins, and a protein called interferon gamma. Those are the two main ones that they make. When we're talking about TNF, there's lots of different versions of TNF. You don't have to know them. Just know that they all can have overlapping functions. And so the lymphotoxins are some members. You can have membrane-bound or secreted TNF. They all are going to bind to the different receptors with different affinities, but they can activate the cells or kill the cells. And so it really depends on which receptors are being expressed. Now, this is a pretty complex set of proteins. They have overlapping results, and it's not really clear how you get differentiation of killing a target cell versus activation of a target cell. But all I really want you to know from this is that they can do either. In general, if you have signaling from TNF receptor 1, you get killing. It's not always true, but it's basically the same pathway as FAS, right? You activate caspase 8, that gives you caspase 3 activation and DNA fragmentation. If you go through the TNF receptor 2 pathway, that's generally thought to be an activating pathway. And so you get activation of kinases and differentiation of the target cell. However, that's not always true. You can also get activation of the killing pathway. All you really need to know is you can have both. And I would focus here on the killing pathway because we're not going to talk about all the different cell activation pathways downstream of TNF receptors. Okay, so the main thing I want you to focus on, TNF is made, signals through a TNF receptor, gives you activation of caspase 8, leading to caspase 3. Right? Simplify this entire thing to just that one pathway. Now, as I said, there's two cytokines that T cells make. One is TNF, family proteins, and the other is a protein called interferon gamma. And if you remember, type 1 interferons, which we all typically call interferon alpha and beta, they sensitize cells to be ready to shut down should they get infected. The interferon gammas also, or the interferon gamma also interferes with viral infection, but it does it differently. Okay, so interferon gamma doesn't cause cell death. It activates cells to get ready to respond to infection. And it does this through some kinases. And what happens is, we've talked about some of these before, the cytokine binding to the cytokine receptor brings in the Janus kinases, or what are called the JAK proteins. In this case, JAK 1 and 2. The Janus kinases, Janus is the Roman god, the two-faced Roman god, that they bind and they'll actually phosphorylate each other. That's where the name comes from. That causes activation of the STAT proteins, which are also kinases. Those come in, they get phosphorylated, and when they get phosphorylated, they form a transcription factor. STAT is signal transducer and activator in T cells. And so JAK 1 and 2 come in, they phosphorylate the receptor that recruits the STAT proteins. Those get phosphorylated and become a transcription factor, and it goes to the nucleus and starts causing expression of other genes. These are interferon stimulated genes that make that cell resistant to infection. Okay, now you don't have to know all of the different proteins that STAT1 starts transcribing. Just know that this interferons are affecting the cells or surrounding cells so that they can be resistant to infection. Okay, so I know we've covered a lot today, but I'm sort of distilling it just to what are the things you need to know. What are the initial activation signals to T cells, in particular the CD8 T cells? So CD28 is the costimulatory protein that allows the cell to start making CD40 ligand, IL-2 receptor and IL-2, right? We should put IL-2 here and the IL-2 receptor. And then how do the CD8 T cells get to sites of infection? And so it's not just the same steps as getting into a high endothelial venule. The dendritic cells will tell the T cell you should be looking for these signals. And then the T cells are expressing a set of selectins, chemokine receptors, and integrins that are favorable to getting into either skin tissues or into mucosal tissues. And then once those CD8 T cells exit from the blood into those tissues, how do they kill infected cells, right? So should know from the perforin granzyme pathway what happens, and from these alternative death pathways, how it's killing cells, and additionally, how is interferon leading to cell resistance to infection, okay? Those are the main take-home points from this lecture. Okay, so this is sort of a step back looking at B cell responses. If you remember, one of the main proteins that's induced by CD40 signals on a B cell is activation-induced deaminase, okay? Now, it's a little different than a CD40 deficient person. Patients which have autosomal recessive mutations in AID have a particular phenotype. In particular, they have massive germinal centers. These are huge. And so the question I have for you in terms of a B cell response, what do you expect the B cell phenotype to be? What isotype? Will you have somatic hypermutation? Will you have isotype switching? Do those occur? And what do you expect the T cell phenotype to be? Is AID important in T cells? Is it expressed? Does it affect the T cell phenotype? And with that, I'll stop and take any questions.
Dr. Blattman.
Yes.
So patients who are deficient in the seroglycine, because it has such a broad role, would that just kind of result in death, or would it just specifically result in possibly CD8 cell deficiency or T cell deficiency, I guess?
So there are no known patients with mutations in seroglycin. The main reason is probably not immunologic, it's probably neurologic. They're not able to send out neurotransmitters. And so it is probably embryonic lethal.
Okay, thank you.
So one of the questions on chat is, would effector T cells be those that are proliferating? Well, yes, sort of. As they're proliferating, they're differentiating into effector T cells. So you have to proliferate to become an effector cell. And really, once it becomes an effector cell, it stops proliferating and is just going out to look for cells to kill.
Dr. Blattman?
Yeah.
So in the FAST pathway, that occurs when there isn't a lot of antigen stimulation. So typically when you get T cell killing, it's because there's an imbalance between MHC self-protein presentation and antigen presentation. Is that imbalance still there, or is there something else going on?
It's not really an imbalance in the MHC. It's just that, in particularly viruses like herpes viruses, are not expressing a lot of protein. And so there's nothing to present, really. You still have MHC, but there's not a lot of viral proteins on the surface.
Yes, so wouldn't there still be the no-kill signal because there's so much of the self-protein outweighing the non-self-protein being presented?
So right, so these are CD8 cells. They don't have a no-kill signal. You think you're thinking of
yes.
NK cells. NK cells would have that. Now, NK cells use the same pathways to kill target cells, but what you're saying applies only to NK cells.
Okay, gotcha.
Other questions? Okay, if there's no other questions, we'll see you on Thursday.